computer. Okay, so just so you all know, we're starting the recording now. All right, so this is part 2 of immune privilege. And March 22nd and March 24th, that's next week, we'll be talking about the ontogeny of mononuclear phagocytes, okay? So today I'm going to focus mostly on antigen presenting cells in terms of immune privilege. So this is going to be more at the cellular and molecular level rather than the previous set of the previous lecture, which is more describing the phenomenon and some of the features that explain the phenomenon. And if you remember, we went over these 3 facets of immune privilege. There are anatomical features such as blood tissue barriers. We didn't talk about too much, but we did talk about pauci-lymphatic nature of the tissues. And we also talked about APCs. This is a form of immune tolerance. And then finally, we talked about immunosuppressive factors at the local level. So we're going to be examining these 3 points to a certain extent now in the context of antigen presenting cells, and we're going to focus on the cornea. So, you know, the cornea is really interesting in this sense because it's clinically evident that immune privilege exists. So just to give you a background of the anatomy of the cornea or the anatomy of the eye, so that you can see in the context of the cornea. You have the ocular surface here, okay? This contains the cornea and the cornea proper. You have the edges of the cornea, which is the limbus. And then adjacent to that, on top of the sclera, which is the white rigid part of your eye, you have the mucous membrane that sort of goes behind here and doubles over under your eyelid. So this is actually bona fide mucosal tissue. And you can see that here are your eyelids. You can see that in this picture here. This is a patient with allergic conjunctivitis. This is similar to what looks like to be pink eye. The pink is really in the mucosal membranes, which is called the conjunctiva. And what's interesting is you have an inflammatory reaction that occurs right adjacent to the cornea, but yet at the same time, the cornea is still very clear and not seemingly clinically involved. If it was clinically involved, this patient would have trouble seeing through the cornea, because the cornea essentially is a window. So if you have inflammation, you're then perhaps affecting the way light enters and light scatters in your eye. And that is the case. So this is a really interesting clinical scenario where you have inflammation at the really adjacent structure, but not in the immune privileged tissue. Another clinical piece of evidence of immune privilege is in the corneal transplantation setting. Here is a allografted cornea in patients. And this is, from my understanding, this is the only full tissue graft organ transplant, essentially, that does not need to be tissue typed in order to for the graft to survive. So in the US, for example, tissue typing, HLA matching is not required and not normally carried out in normal risk grafts. It's not to say that they're not under corticosteroid treatment, but in other transplants, you need both tissue typing and a massive corticosteroid treatment in order to maintain survival. In
As
mice,
I understand it, I think actually liver transplant, they do not type until post-transplant. It's all blood type for blood type only matching.
Thanks, Carl. That's interesting. And actually, I think the liver is considered immune, is considered to have immune tolerance or immune privilege to a certain extent.
Yeah, it's tolerogenic.
Yeah. Thank you for adding that. So and I don't know about in mouse models, but in mouse models with cornea, you actually get 50% spontaneous acceptance. This is without steroids, and in certain donor recipient combinations. And so this is showing an accepted graph where you can see light actually penetrating, whereas this is a rejected graph. You can see the light. This is a slit lamp, which the clinicians use, and you can see it doesn't pass through the rejected cornea. And with respect to one of the features which we said, immune tolerance, again, it's not this is work done by Sunil Chauhan. This is I believe 2009, was published in JI. You can see in this spontaneous acceptance versus rejector, you can then look into the lymph node. And what he found was that in accepted mice, they had an increased number or increased expression of FOXP3. So that's a marker of T regulatory cells. Whereas in the rejector, you can see that this is substantially lower. And they went on to show that these Tregs are actually allospecific suppressors of effector T cells. So there is a form of immune tolerance. So that's checking off our list of systemic immune tolerance initiated by the graph. Just as a side note, we went through the list generated by Rupert Billingham of immune privilege sites as well as tissues. Remember, a site is where immune privilege has shown to be existent with an allograft. A tissue both is an immune privileged site, but it's inherently immune privileged. That is to say that if you take that tissue and put it in a non-privileged site, it still enjoys privilege. So this is an example published by Geimer and Mendel in 1994, where they compared cornea allografts, pancreas and skin in the subcapsular, the kidney capsule. And you can look to see what they did was basically have a graph score of the higher the graph score, the more the graft is I wouldn't say functioning, but at least histologically appearing relatively normal. And you can see that that holds for 21 days or so, and then slightly begins to fall off. Whereas if you look at the other two tissues in the kidney capsule, the graph score seems to fall off quite quickly. If you look at the infiltration score, there's a slow trickle of infiltration over the time course, whereas in the other two tissues, you can see there's infiltration happening quite readily, quite quickly, signifying that this is an immune privileged tissue with respect to the cornea. Okay, so what's going on? So sort of to tease apart what might be going on, we can actually think about the immune reflex arc. So if you remember, this is where the afferent limb picks up antigen by way mostly of antigen presenting cells that migrate and get homed into the lymph node where the T cells are, and they reach and integrate or interact with their cognate T cells with the T cell receptor, where they then expand, differentiate, and peripheralize into the blood, where they can make their way back to the inciting antigen. In this case, this is just modeled in the skin, but you have the same thing in the cornea. Now, with respect to the players in these two limbs, again, the antigen presenting cells are carrying antigen to the lymph node. And whereas in the efferent limb, it's mostly the primed effector or helper T cells that are peripheralizing and going back. So we can focus on these two cell types to get an idea of what's happening here in both sides of the arc. Now, just to remind you, this is to check off the tissue barrier in cornea in terms of the facets of immune privilege. The cornea in the steady state is avascular, the cornea proper, but just adjacent to that, you have lymphatics. So you have both lymphatics and vessels. So I'm kind of using this as a double-edged sword here. On the one hand, it is a posse lymphatic state, but on the other hand, it's not that it's completely inaccessible to both processes, both limbs, the efferent and afferent and efferent. And furthermore, as you can see, in an inflammatory state, you do have invasion of these vessels into the cornea proper, which can amplify the trafficking of these cells. Okay. Now, if we're talking about the efferent limb and wondering whether the efferent limb is contributing to this immune privilege, one way to do that is to do an adoptive transfer. We discussed a little bit of these experiments in the prior lecture. So what you can do is take primed allospecific T cells from mice that had previously rejected their graft and transplant them into a naive, freshly transplanted recipient mouse. So now you're basically you're stepping over the question of the afferent limb. This is not now has nothing to do with the afferent limb. You're specifically asking, is there an accessible efferent limb? You know that there's blood vessels there in the limbus, so that means that potentially T cells can infiltrate the cornea. And so and this is one paper just to demonstrate that the efferent limb is indeed accessible or plays a role. There are other papers that have done this sort of experiment, but essentially they did exactly this. D6 is a gene specifically that they looked at as a decoy receptor. That's not really relevant to this talk. If you look at the D6++, you can just consider those wild type T cells. These are taken from primed immune rejected animals transferred to a freshly transplanted mouse. So you can see with this, in this group, you can see almost looks like full rejection within by day 20, whereas if you didn't transplant any T cells at all, you get 25% survival. This is a bit lower than what we're used to, but you can see that there's a statistically significant difference between the two, arguing that the efferent limb, again, is intact. So the
question for the alloreactive T cells, are those like harvested from an MLR, or how are they identified and harvested for?
Yeah, so what they did was to take a rejected mouse and simply pull the lymph nodes, purify the T cells. I think this was done by CD90 Phi1, let's just put it that way, and then transferred into the recipient mouse intravenously. And that does the trick. Yeah, no need for further expansion, just straight harvest, purify, transfer.
Thanks.
Mm-hmm. Okay, so going into the afferent limb now. And remember, we're talking about antigen presenting cells, at least one major aspect of this. If you think about antigen presenting cells, and we'll get into this on Monday, more specifically Monday and Wednesday, more specifically, but just to give you a sort of broad perspective, the types of antigen presenting cells we have: macrophages, monocytes, dendritic cells. These are all slightly different. They present antigen in different ways. I think it's clear, it's without going into too much detail, it's quite clear to say that dendritic cells are really the T cell machines, T cell priming machines. They're made to quickly process antigen in a manner to be able to specifically present through MHC class 2 with co-stimulatory molecules so they can activate naive T cells. If you look here in the immature state, they have lower antigen expression on the cell surface. They're expressing MHC within the cell itself, which is how they get loaded. But at the cell surface, doesn't become highly expressed until they become mature, when things are loaded. And at that point, sorry for these arrows, they've moved over, but antigen capture goes down, antigen presentation goes up, and co-stimulatory molecules go up. This allows them to now go to the lymph node in a CCR7 dependent manner and access T cells and find their partners, their cognate T cell receptor T cells, to then expand these T cells. So I think we can argue that we're mainly talking about dendritic cells in this setting in terms of antigen presenting cells activating a T cell response in the lymph node against the graft. So so this is an interesting topic because, you know, early on in the 70s, 80s and even into the early 90s, there was something curious about the cornea that was picked up on, which I'm showing you here. If you were to stain for class 2, MHC class 2, whether it's IA, IE, or both, what Niederkorn et al. Are showing here is that in the cornea, you have very few. So this is a cutout cornea. I believe it's probably a mouse. Here is the limbus, where in A, where you see the density of these cells. And then you're moving towards the central cornea, and you can see they're few and far between in terms of MHC class 2 positive cells. And this is compared to the conjunctiva, where you can see these in relatively robust numbers. And this is similar to what one would see in the skin. So the question, or the theory at the time, the hypothesis at the time was probably that there was a lack of immunocompetent cells antigen presenting cells that could launch an afferent response. But that turned out to be not the case, at least with respect to there not being cells in the cornea, antigen presenting potential cells. So if you look, this is now, now that here's in the 2000s, just to show you, but this is probably not the first paper, but just to demonstrate. Now we come to the time where we've extended our antibody staining repertoire. We're looking at CD45, which is a common leukocyte antigen. Looking also at F480 and CD11B, CD11B being a myeloid cell marker. This is data from the paper. Here you have the cornea. You can see in A, you can see the edge, which is where the limbus is. And now staining for CD45, you can actually see more abundant number, a higher abundance of cells. So it's not like the cornea is absent. And you can see that the number actually goes down as you go towards the central cornea. But again, these cells were not absent. Just looking for MHC class 2 made it appear as if there was an absence of antigen presenting cells. But it turns out that these cells are there, yet they're not expressing MHC class 2, which is relatively irregular when looking at non-privileged tissues, such as the skin, where you can readily find MHC class 2. At the time, they're referred to mostly as Langerhans cells, which is true for the skin. Here's another paper in 2002, looking more at the different depths of the cornea, but essentially, with the question of whether there are dendritic cells there, which are really the the best antigen presenting cells in that sense. And CD11C for a long time was considered a gold standard marker for DCs. We now know that not to be the case. But what Hamra et al. Showed is that there are indeed CD11C positive cells, suggesting that there were in fact dendritic cells, but that again, these DCs are peculiar in that they don't have cell surface expression of MHC, at least not 100% of them. Here you can see what he's calling a CD11C positive cell, which has minimal MHC class 2. And what they showed in the deeper layers of the tissue is that there are other macrophages there, and many of them are not expressing class 2 as well. The other argument was that because the cornea, for example, in the epithelium has tight junctions, maybe the problem was that the antibody was not really making its way into the tissue when they're doing immunolabeling. So now we're in the EYFP CD11C knock-in phase, or I guess time frame of experimentation or toolboxes. This is a CD11C YFP knock-in. So this is a mouse, clearly. And you can see quite beautifully, if one was to argue that you need CD11C cells to show that there are dendritic cells in the cornea to say that there's immunocompetent antigen presenting cells, clearly there's plenty. Of course, you can see there's a higher density in the limbus, but as you move towards the middle, they're there too. My AirPods might die, and I might have to switch. If my voice goes off and I seem to be talking to myself, if somebody can just pop on the video and let me know. Thanks, Carl. Okay, so what you can see clearly here is YFP cells, less so in the center, more so in the peripheral places of the cornea, and you can see in a cross section they're clearly there. You can see there's an abundance of them in this subepithelial space, interdigitating the epithelium and some in the stroma. So you can't argue that there's, at least with CD11C, that there's no dendritic cells in the cornea. In fact, this, I'd mentioned Langerhans cells before. My intention is not to get too specific into the question of Langerhans cells, but there was a time in the late 2000s where Langerhans cells, or Langerin, which is a protein expressed with Langerhans cells, we don't fully know what it does, was a hot topic because they had showed that there was Langerin positive dendritic cells in the skin, in the dermis, and that were inappropriately or inaccurately being called Langerhans cells. Anyways, to make a long story short, they came up with several models to look at that. And we had showed that there is indeed Langerhans cells in the corneal epithelium. The curious thing about them, though, again, if you look at Langerhans cells, this is Langerin expression in the epithelium. You can see there are some that express MHC class 2, but there are those that do not. So even if you're focused on this classical Langerhans cell, and in the next set of lectures, we'll actually talk about the ontogeny of these cells, which is also very interesting. But suffice it to say, going back to this, that there seems to be this abundance of MHC class 2 negative cells at the cell surface expression that may explain the lack of an afferent limb in corneal transplantation. And it turns out that the same phenomenon occurs in humans. So this is work done by Nickelbein et al. At UPitt, I believe, showing this is a cornea. This is a wedge of a human cornea that was removed for staining with CD11C and MHC class 2. I believe, I should have marked this, but I believe MHC class 2 is in red and CD11C is in green. So you can see there's plenty of CD11C positive MHC class 2. And by the way, this should be HLA-DR, which is the human leukocyte antigen for humans, not MHC is in mice. But you can see there's clearly an abundance of dendritic cells that do not express HLA. Again, signifying that the mouse work is relevant in the human setting as well. So let me stop here and entertain any questions if there are any. Any questions so far? Okay. So let's move on. So what I've shown you so far is that we have an abundance of these MHC class 2 negative, and I say cell surface because it's important to differentiate. Just because what I'm not trying to say that they're not expressing MHC class 2. What I am trying to say is that the MHC class 2 simply isn't on the surface of the cell where it should be antigen presenting. So those were all descriptive studies, and now you sort of want to ask the question whether there's a functional impairment based on this this appreciation. So what's nice about the cornea is that you can do corneal transplant studies. We know that there's something called passenger leukocytes. When you do a transplantation, you're not only transplanting the tissue, but you're also obviously transplanting the cells in that tissue. And in the case of the cornea, like many other tissues, you have antigen presenting cells housed in the tissue. And those cells themselves can travel to the lymph node and trigger a T cell response against the tissue. And as Carl had mentioned before, you can actually do this in a dish. It's called a mixed lymphocyte reaction. It's actually a quite strong response. So what you can do is now take a donor tissue and with the antigen presenting cells, and you can ask specifically whether these antigen presenting cells are functional. That is, do they make their way to the lymph node and trigger a T cell response? Or you could just specifically look at a T cell response. And the way to look at a T cell response in this context is, at least at this time in the early 2000s, was clearly just by looking at immune rejection. So what Huck et al. Did in this experiment was to take an MHC class 2 knockout allograft. Okay, so only the allograft is MHC class 2 negative, and compared it to a wild type. Remember that I said previously that in certain combinations of donor recipient, I believe this is B6 to BALB-C, you get a 50% spontaneous acceptance of the graft, and you can see that here. So in the normal risk graft bed, and I'll explain that to you in a second, you get a 50% spontaneous acceptance. If you transplant the allograft, an allograft with a knockout for MHC class 2, there's no difference. So you would expect that that if MHC class 2 was playing a role, that there would be some increased survival perhaps to this level, but there's not. So what it's suggesting, at least at this point, is that there seems to be no functional role, at least in the context of graft rejection, for these donor antigen presenting cells, suggesting that they're not functional. That's quite a complex topic. I wonder if anybody has any questions before I move into the high risk graft bed.
So would the MHC class 2 have to be presenting self-antigen from the allograft in order to elicit an immune response or a rejection response from the host?
Yeah, so it's not necessarily, so it's an interesting topic, and it's not necessarily that they're presenting self-antigen, it's that they are expressing MHC class 2 themselves. And the MHC class 2 that they're expressing is allogenic, right? Does that make sense?
Yeah, that makes sense. But with I see.
So it doesn't,
yeah. Independent of the antigen presenting. I see what you're saying.
Correct.
Okay. All right. That makes
Correct.
sense.
From
Gotcha.
what in my understanding, and these are really powerful responses. So you have, without getting too deep into transplantation immunology, but you have two ways in which allo recognition occurs. One is direct, which is this: the passenger leukocytes travel to the lymph node, and they themselves activate a T cell response. That is a really robust rejection pathway. The other is indirect, which is less robust. Indirect is the host APCs actually go into the graft and pick up alloantigen, non-self antigen, and travel to the lymph node themselves and elicit an immune response. So there's two types.
Gotcha. That makes sense. Thank you.
Okay. Now, in this context, we're only looking at direct. We're only looking at donor direct antigen presentation, simply for the fact that we're asking a specific question of whether these antigen presenting cells might be functional or not. Okay. All right. So let me move on to thank you for the question. Let me move on to high risk. So high risk is a completely different setting. It's made to model high risk transplants in the clinic. These are patients that come in with an inflamed graft bed, and what clinicians try to do is quiet the inflammatory reaction that's occurring in these patients prior to doing the transplant. But in many cases, they're able to do so, but there's still facets of the graft bed that make it high risk. To model this, what you do in mice is to essentially trigger an inflammatory response in the cornea prior to the transplantation. And there are different ways to do this. The way that was used here was through putting actual sutures, penetrating sutures into the cornea without a graft for 2 weeks. And these sutures alone will trigger an inflammatory response. You then, after 2 weeks, you then remove those sutures, and then you remove the central cornea so you can put a fresh graft on. But remember, the graft bed is now inflamed. And in that setting, you have what I'm showing you here, complete rejection. Okay. So in this setting, you can now ask the same question. If you put on a donor tissue that is MHC2, has a genetic MHC2 knockout, does it affect the fate of the graft? And here, quite clearly, you can see a big difference. The MHC class 2 knockout is surviving at a higher rate. Okay, so about 20% of these grafts are spontaneously surviving. So there you see a key difference now. What that is suggesting is that there is a role for MHC class 2 donor APCs in this rejection setting, therefore telling us that these cells can be immunocompetent in an inflammatory setting. Okay.
Quick question.
So it's situational, in other words. Yeah, go ahead, Carl. Carl, can you hear me okay?
I can. Can you hear me?
Right. Yeah, I can hear you fine. Thanks.
Yeah. What's the capacity of the corneal tissue for healing? Is it like pretty minimal, or?
I'm not sure. So if you were to just wound it?
Yeah, just like a minimal wound or a scratch, like does the tissue heal pretty quickly? And I guess I'm asking in the context when you have a rejection event occurring, is the tissue healing while it's rejecting?
So yeah. So let me first say that I'm not a clinician, but so I guess it's context dependent. So if you were to just do an abrasion, the cornea would heal pretty rapidly. The epithelial cells, just like in the skin, would cover kind of a superficial wound. That's not an issue. You know, as you know, we do get scratched. Our corneas do get scratched. Now there is the case that if it's a deeper wound, there might be a scar, and if the scar is in your axis of vision, then it is problematic. So there are situations where it can't heal. From what I understand in the clinic, when the patient is undergoing a bout of immune rejection, they really what they do is essentially they increase the corticosteroid dose, and that usually takes care of it. But you're right, a number of if the patient experiences numbers of bouts of immune rejection, it could get to the point where the survival rate is now the graft is rejected to a certain extent.
I guess, does that wound healing process, like how quick or how slow it's occurring, does that interface with the sort of tolerance here? Like, is part of what you're measuring in tolerance actually wound healing?
I'm not sure. The thing, and I'm sort of thinking out loud here, so if you permit me, the thing is that immune tolerance. Still hear me? Yeah, there it goes. Can you hear me, Carl?
Yeah, I can still hear it.
I switched up. The thing is that the immune tolerance is a antigen specific adaptive process. Wound healing is not. So there are essentially two different processes. That's not to say that the wound itself may or may not affect immune tolerance. Maybe, I don't know, maybe if you phrase your question a little differently, I can answer it. I'm not sure how else to.
I guess in terms of percent survival, I wonder if that measure is a sort of combination of both the allograft capacity for wound healing and or graft bed capacity for wound healing and the rejection process.
So maybe this will help. So in mice, this is relatively standard. So the way it works, and you might be, I think maybe you're getting at also how accurate the graft is placed, because that can affect it. So the way it's done is when you do a transplant, if you have any problems with the graft within the first week, that can be identified, and then that mouse will not be counted. Basically would be an outlier because it's not rejection. Immune rejection takes a week or so, if not 2 weeks in the cornea. So you remove any outlier that there's technical failure or some problem with the cornea, maybe prior to that the investigator didn't see. Okay. And in this situation, you're just basically following out the mouse. But it's interesting because I've been involved in these experiments. Sometimes you do see that there's an inflammatory response. We don't know whether it's T cell at the time because we're just actually looking at the graft visually. You can see some swelling, and sometimes that subsides. So I'm not sure if that helps.
It does. I think in my time in liver transplant, I used to think of tolerance in patients as a process that is both like a balance of the liver's capacity for self-regeneration and injury occurring by immune rejection.
Well,
and I wonder if this is like a similar thing here where you can have like a low-level rejection of the allograft that is subclinical because of healing.
Right. So there's a key difference between the cornea and the liver. There's no regeneration of the cornea. So in the cornea, the critical target in rejection is the endothelium. So the most posterior layer of the cornea are these neural crest-derived endothelial cells. These are not vascular endothelials. And what those do is help to pump in nutrients and pump out water. Those are postmitotic cells. So as those die, essentially you need a certain threshold number of those for the cornea to maintain itself. And so as those die and you reach some threshold where the cornea cannot maintain itself anymore, it drops out. So I suspect that the liver is somewhat different, that regeneration capacity is there, whereas in the cornea it's not. So, but I do think that you're right, that probably there is a balance between some innate wound healing response versus the adaptive response, and they do intersect, but I don't know. I don't have a specific answer. Yeah.
Well, thanks for the informed speculation. I appreciate it.
Yeah. Thanks for the question. So just to wrap up this slide here, just checking my time. What the high risk situation tells us is that that the donor APC can play a role, but only in a specific situation. They can be functional, but only in a specific situation. That is where there's a level of inflammation that may or may not activate them. Now, to take this one step further, we talked about the 3 facets of immune privilege, one being tissue barrier. We covered that in terms of the cornea being polylymphatic. Another one is immune tolerance. We covered that. I showed you that Tregs are activated by way of looking at the lymph node and doing suppression assays. The third point is whether there are local immunosuppressive factors. In the previous lecture, I talked about it mostly in the case of the efferent limb. Now I'm going to talk about it in the afferent limb, just with this example here, which is thrombospondin. Thrombospondin is a matricellular protein. That means it gets placed in the extracellular matrix and actually has a function in the extracellular matrix, which is not the focus of this.
It's also expressed and maintained on cell surface, and it can act, for example, to activate latent TGF-beta and other such things. It's been shown that thrombospondin is part of the immunoregulatory process. One of the interesting things that came up with thrombospondin through work by Marika Sarfati in the early 2000s was that thrombospondin seemed to play a critical role in antigen presenting cell exhaustion, and this had relevance with MHC class II expression. So what we did is we simply looked at thrombospondin knockouts to look at MHC class II expression, and you can see that that number is quite high, or higher when you look at compared to the wild type. So you can see 12%. This is MHC and co-stimulatory molecules, and so you see it's elevated in all of these, and as well as the cornea. So if you look at a normal cornea, you can see the elevation. So using that same sort of technique, you can graft now an allogeneic thrombospondin knockout donor onto a wild type allogeneic host. And when you do that, one finds that the thrombospondin knockout donor grafts reject at a much higher frequency, suggesting that this is playing a role in the resistance to MHC class II expression and mediation of subsequent activation of T cell responses in the lymph node that then mediate T cell graft rejection. I don't want to get into this. Carl, you might enjoy this maybe later on after class, but this is an ELISPAT assay to look at indirect versus direct forms of immune rejection. And it turns out that the indirect, based on this assay, was not statistically significant, whereas the direct allorecognition was increased, which is consistent with the conclusion that donor APCs with MHC class II expression can activate T cells, and that the MHC class II expression on these donors, if through natural inhibition or resistance, is playing a role in preventing the afferent limb from doing its job. So just to sort of conclude that part, there is an inherent expression of thrombospondin that is playing a role in suppressing MHC class II in the cornea, which is relevant to what was found here. So this is my last slide. I'm just going to wrap up, and then we may have time for questions. I'm going to put this in the context of corneal immune privilege, and then which touches upon really both the last two lectures. So the cornea, as I mentioned, it presents or possesses all three facets of immune privilege: anatomical features, again, pauci-lymphatics, immune tolerance with Tregs, and immunosuppressive factors. The example I gave was thrombospondin. It's important to show or to appreciate that this corneal immune privilege is clinically evident. I showed you the example of allergic conjunctivitis, where the neighboring tissue is wildly swollen, whereas the cornea remains transparent and uninvolved. The other, of course, what we focused on mostly is that corneal allografts can survive in the absence of HLA matching, and clearly there's the liver that seems to be similar in that way. Contributing to this IP, so we talked a lot about the afferent limb of the immune reflex arc. That seems to be limited. And the evidence that I showed you today is in part to an irregular abundance of cornea APCs with an absence surface expression of IAIE. Remember, this MHC class II is critical for presentation to T cells in the lymph node. And then in contrast, there is evidence of an accessible efferent limb. So that's the other side of the reflex arc to the cornea. And I showed you evidence of that with adoptive transfer studies of primed alloreactive T cells, which increase the rates of rejection. Okay, so we have a few more minutes. If there's any other questions, please feel free to ask.
I have one question. So in terms of thinking about, I guess I'm wondering, like, is there a physical barrier between the limbus and the corneal parenchyma that would prevent activated, antigen presenting cells from being able to migrate into that tissue? Or is it more that because that limbus just has lymphatics, it's I guess preferentially exposed to things that could cause inflammation, whereas the corneal parenchyma, requires like angiogenesis and development of these lymphatics to be exposed to these same stimuli.
So I think that's a good question, and let me see if I fully gathered. So you have so I think the simple answer is no. I don't think there's really a true barrier there. But even if there was a barrier, I don't know that that would prevent it. So there's certain barriers that are really strong, and there's certain barriers that are, maybe it's not that they're strong or not, but antigen presenting cells can surpass them quite readily. So if you think about, for example, there are barriers everywhere. Think about the epidermis and the dermis. There's a basement membrane there that Langerhans cells that live in the epidermis, actually are intraepithelial, must cross in order to get into the lymphatics in the dermis. So, and you have the same situation in the cornea. You have antigen presenting cells in the intraepithelial space that for them to get into the lymphatics, they need to travel to the limbus and then get into the deeper parts of the tissue in order to get into the terminal lymphatics. So I'm not sure that is a really robust barrier, if you know what I mean.
Yeah, yeah, that makes sense, I guess. So what so is it then that you don't, I mean, because you do see expression of like MHC class 2 at least in the limbus antigen presenting cells. So I wonder why you like don't see maybe more migration of those cell populations into the parenchyma that could lead to rejection or something like that.
Yeah. So you're okay. So you're talking. So okay. Think about how the cornea looks and structure of the graft. The graft would be in the middle, right? It wouldn't include the limbus. So those guys that probably you would consider immunocompetent at the limbus because they have some MHC class 2 expression at the cell surface. Those can migrate into the donor, pick up alloantigen, and then migrate out. Clearly, this is a lot longer of a process to an extent when you compare it to the passenger leukocytes, which are already there, and all they need to do is just migrate out. You see what I'm saying? So I didn't really talk about that indirect pathway. That pathway is there and active. It's just not as strong as the direct pathway.
Okay, that makes sense.
Okay. We can talk about this more if you want to later. You can feel free to email me and we can talk about it more if I didn't fully satisfy your curiosity. I don't know if there are any other questions before we wrap up.
In corneal transplant, is there antibody mediated rejection?
No. I'm not going to say that it's completely absent, but I don't think it's really the clinically significant problem, let's say. What we know in corneal transplant rejection is it's mostly CD4 mediated. Even CD8 killer T cells are relatively are not deemed important for immune rejection.
Okay.
All right. Again, I guess our time is up. You have my email. This will be posted through the Sakai website, so you have access to it. And feel free to reach out to me if you have any questions. Okay. Thank you.
Thanks.